Jackson in the Department of Ecology, and as you know, she's arrived last year, and it's been fun to have her as a neighbor in Johnson Hall. Doug got her bachelor's degree at Berkeley, and then went to CU for her PhD, where she worked on water uptake in subalpine forests. And then she did a postdoc at NCAR, working in Tibet. Was that the Tibet project? And then from there went to University of Sydney, where she was a research and education fellow there. And her interests really are on water uptake and what that means for carbon sequestration, I guess, and how climate change is going to affect that. So take it away.
All right. Thank you, Kathy. Can you hear me okay? Okay. All right. So I'm going to be talking about work from my PhD to my postdoc, and also what I'm doing these days. And I sort of just want to take you on this journey of my past research interest and sort of how I've arrived to where I am now. And so this is what I knew about plants going into college. I sort of knew that they needed water and nutrients and light, but it wasn't really until I worked for Todd Dawson shortly after graduation on redwood tree water uptake that I truly began to appreciate just how amazing plants are in moving water around. And so we were looking at how redwood trees were able to move water from the roots all the way up to the canopy. And one of the things that we had found out in this project is that these trees can actually absorb fog directly through their leaves. And so if you look at the morphology of the needles going from the bottom to the top, you see it changes a lot. And the upper needles essentially act like sponges. And so they could take up the fog directly through the needles and alleviate some of that the tension. So that really got me interested in looking at plant-water relations. And so I did my PhD in Colorado working at the Nightwall Ridge Ameriflux site. It's an eddyflux site. And so I came into this project when they had 5 years of data of CO2 and water fluxes at the ecosystem scale. And they had some interannual patterns, but there was really no way to explain what was going on. And so they really needed someone to come in and look at some of the mechanisms to explain some of these patterns. And so I looked at carbon and water relations from a plant perspective for my PhD. And then shortly after that, when I did a postdoc, and I did some work in Tibet, but I also worked for 2 summers in a Ponderosa Pine forest in Manitou Springs, and that sort of helped shape me think about how different this ecosystem was using water compared to that ecosystem. And that really sort of helped me arrive to where I am now. So this is a picture of Lubrecht. And prior to working at Lubrecht, I was really viewing water from the starting point of where the trees were just getting the water and how that was moving through the tree. I wasn't really thinking about how water moves through the landscape before it actually reaches the tree. And so I've been working with Kelsey Genso, who's the new assistant professor at University of Montana, and he's a watershed hydrologist. So he's really helped me bring that perspective into my research, thinking about how landscape really drives these patterns of water use. And this is a picture that I took a couple weeks ago when I was out there. You can see the yellow. The yellow is Western larch. It's a deciduous conifer, which is quite rare, but you can see it across the landscape, and it's really amazing because it's sort of it's a great example of how the landscape determines where species grow. So larch tend to grow on northern aspects where there's a lot of water. And so you can see in the fall, just really clearly, these areas that larch tend to do really well. So why am I interested in working in the Western U.S.? Well, we know that in the West, there's relatively high carbon uptake in these high elevation montane ecosystems. And if from the east, in the eastern U.S., we know that's quite common. The rates are quite high because of the forests that are coming back after they were deforested hundreds of years ago. But the West is really dry compared to the East. And so these islands are really, they're isolated to higher elevation ecosystems. And so in this one paper from Dave Schimmel, they found that over 70% of annual GPP occurred in elevations over 750 meters. So it's really sort of orographic effects that bring moisture into these high elevation ecosystems that are surrounded by otherwise semi-arid and arid ecosystems. And we also know the climate in the Western U.S. has changed quite a bit in the past 50 years. And so this is a study by Philip Moat, where they used both snow-till observations and modeling to show that across the western United States, snowpack has decreased significantly in the past 50 years. And we know that in these high elevation ecosystems, snowpack is really important for productivity, and so further declines in the snowpack really has the potential to decrease productivity in these montane ecosystems. Okay, so here's just an outline of what I'll be talking about today. First is looking at the importance of snowmelt and productivity in a subalpine forest, which is my PhD work. And then the second is looking at the importance of rain in a Ponderosa Pine forest. And then going into the project that I'm working on currently, which is climate impacts on forest ecohydrology from the hydrological and physiological perspective. Okay, so to address the first question, the initial question I had coming in for my PhD was looking at how do changes in precipitation dynamics, that is less snowfall, affect carbon uptake rates. And as I mentioned, I was working at a flux site, and so I had a lot of eddy flux data to use. And so I used about 9 years of eddy flux data to explore some of the natural variability in precipitation, and then looked at how that affected carbon uptake rates. And this was at the Niwot Ridge Flux site. It's a subalpine forest. The dominant species at this site include Engelmann spruce, subalpine fir, and lodgepole pine. And this is just showing you what the NEP data look like from Niwot Ridge all 9 years. This is looking at it from the perspective of the biosphere. So unlike Ash's talk last week, I'm an ecologist, so we're looking at it from the perspective of the atmosphere and not of the biosphere, not the atmosphere. So positive values mean carbon that's being taken up by the forest, and negative is carbon being released by the forest. And so you see for 9 years this pattern where you've got respiration happening in the winter months, and then here you've got spring turning on the forest, and the values become positive. And at some point, this value crosses 0. So all the carbon that's sequestered by the ecosystem is the area under the curve up there. But the other thing you'll notice from this is that every year, the point at which this this upslope happens is quite variable. And so I wanted to explore some of those patterns. And then there were a series of papers that came out about 10 years ago that were looking at what effects warmer temperatures had on productivity. And so they were looking at the relationship between warmer temperatures and growing season lengths. And so this is a paper by Trakina where he looked at 28 different flux sites across the United States and looked at how the carbon uptake periods, the number of growing days, affected the net ecosystem productivity. And they found this positive relationship. And so the longer the growing season, the more carbon uptake by the forest. But what this paper did was they took 28 different sites. They didn't follow one site across time and look at the variability that one site might have. And so I wanted to sort of look at this relationship, but look at it from Niwot Ridge. And particularly, I found it interesting there because it's a conifer forest. And so these trees have their needles on all year long, and they're able to photosynthesize even when there's a significant snowpack on the ground. And so when I looked at this relationship using 9 years of eddy flux data, I actually found a negative relationship. And so as you increase the growing season length, you actually get a decrease in net ecosystem productivity. And I found that this was largely related to the size of the snowpack. So this is snow water equivalent. And so if you actually have a long growing season, you actually have a shallower snowpack compared to a short growing season and a very large snowpack. And so we know that snowpack sort of drives NEP in this ecosystem. But I wanted to ask further, can we quantify how much the ecosystem is relying on snowmelt for ecosystem carbon uptake? And my approach here was to use isotopes to trace water source, and then use an ecosystem process model to partition the GPP into the snow-contributed versus rain-contributed GPP. So just a very brief review of stable isotopes. These are just a percentage of the different isotopes that are out there in nature. And the ones that I'll be talking about today, it's just hydrogen and oxygen right here. And so the notation is just this delta notation, which is just the ratio of some sample by the standard -1 to get this per mil unit. And so everything is just expressed in delta notation. So everything is just relative to a standard. That's how these values are expressed. So it turns out there's actually a really nice relationship between the oxygen and the hydrogen isotope ratio, and this is called a global meteoric water line. And what you find is that global precipitation tends to fall on this line, but there are some general patterns into where precipitation falls. And so if you have colder regions, so higher latitudes, these values tend to fall out here versus warmer regions closer to the tropics tend to have precipitation values that fall over there. There's also seasonal differences. So if you have snow versus rain, snow tends to fall in this area while rain falls over here. If you come off this line, so your slope is less than 8, you tend to see a lot of evaporative enrichment. And so closed, large basin areas, their precipitation tends to fall off. And so from NIWAT radiation, when I looked at the snow versus the rain, you can see they fall in this area that I would predict. So the snow is very negative compared to the rain, which is a lot more enriched. And so at NIWAT, I went and collected over 3 years soil samples, tree xylem samples, snow samples, rain samples, creek samples, every groundwater samples, just every sort of water source I thought the trees could access to look at where they were using water. And this is just a really classic paper by Jim Ellringer showing how isotopes are used to partition water use. And this is a study where he was looking at different plant function groups growing in a desert in Utah. What you see here is, here's summer rain value, isotope value, and here's the winter rain on the groundwater. And you see that the annuals and the succulent perennials tend to get most of their water from the summer rains, whereas the deep-rooted perennials primarily get their rain from deeper groundwater. And then in between, some plants use a little bit of both. And so this is the approach I was using to look at water use among the different plant species. And so here, just the results for 1 year, just to show you what these numbers look like. In the table, you'll see that groundwater, just look at the deuterium values, the groundwater and the creek water are very similar. What this is suggesting is that the creek water is being, the groundwater is contributing to the creek water, so water is going down and out into the creek. The snow in the ground also quite similar, which shows that groundwater is recharged from snowmelt. And then the rain value was quite a range across the summer, going from more negative to less negative, which is really typical as you have warmer convective storms happening during the summer. And then the xylem value was some range in between that. And so to do this water use partitioning, I used a 2-end mixing model. It's very simple. It just says that the xylem water isotope value is some fraction of rain and some fraction of snow. Okay, so here's just showing you 3 years of data. I've got the rain in the soils. So the upper soils in the yellow circles, you can see they track the rain values really well. Whereas the deeper soils, from 30 to 35 centimeters, they don't change too much during the growing season. They largely reflect groundwater, with the exception of this pretty large rain event that happened in 2006. Okay, so what do the xylem numbers look like? So using that mixing model, I partitioned water use between snow and rain, and you see that for every year, even into October, about 50% of the water in the xylem still had a snow melt signature. This is about 4 months after the snowpack is already gone from the system. The plants are still using a water source that has a snow melt signature. So this is telling us that snow is really important in this ecosystem. So the next thing I want to do after doing this partitioning approach was looking at the ability to quantify how much of productivity was due to snow melt versus rain. And so I collaborated with Dave Moore, who is now at the University of Arizona. He was a modeler. He was working on the SIPNet model for Niwa Ridge. It's called the Simplified Photosynthesis Evapotranspiration Model. And just very basically what SIPNet does is it simulates eddy flux observations and uses maximum likelihood to predict outcomes. So what it does is it partitions this net ecosystem productivity into gross primary productivity and ecosystem respiration. And this is what we're really interested in doing. And then we use the isotope partitioning of rainwater versus snow water to calculate the fraction of GPP that was snowmelt versus rain contributed. So when we combine SIPNet with the isotope, this is what we found. So this is saying that total GPP in that year, 67% of total GPP was driven by snowmelt that year, and up to 77% in 2006. Now with this figure, it's not saying that the trees don't use rain, it's just saying that when the forest is the most active and the most productive, that's the time period that they're heavily dependent on snow. And there have been some independent measurements at the site looking at photosynthetic rates of these different species, and 10 degrees is about the optimal temperature. So you can see it's really these cool winter periods when there's abundant snow melt around that the trees are doing most of their carbon uptake. Okay, so why don't the trees use more rain? Well, Reynolds and Knight, they found that rain events less than 10 millimeters in a subalpine forest, it was either intercepted by the canopy or absorbed by forest litter on the surface floor. So I went through and I looked at all the rain events for all 3 years, and I looked at the average size of the rain events, and I looked at the percent of rain events that were greater than 10 millimeters. And I found that, only about 10% of the rain events were actually big enough, probably, to get into the soil and for the trees to access. And so it might not be that the trees aren't, they don't want to use more rain, it's just that these rain events aren't large enough to actually reach the roots. So conclusions from the first part, I found that years with a shallow snowpack resulted in lower annual NAP by extending the length of the growing season. And the trees do not appear to rely heavily on monsoon rains because many rain events do not actually reach the roots. And then SIPNet demonstrated that about 70% of the annual GPP is snowmelt contributed GPP, and most of which occurred during the early part of the growing season. Okay, so moving on to my second question, looking at the importance of rain. So after my PhD, when I was working in Tibet, I was working in this forest, and this is NCAR had a experiment going on at the Manitou Experimental Forest looking at VOCs, and they had a flux tower there. And so I was working on a project collaborating with David Noon and his students on a way to use isotopes to partition evapotranspiration. So this is a Ponderosa Pine forest. The elevation is about 8,000 feet. And one of the things I was really interested in was looking at how Ponderosa Pine used water compared to how subalpine trees use water. And I remember I had done some reading before working here that Ponderosa Pine were able to use rain when it is actually available. And so this is a paper by John Roden and Jim Elringer where they looked at Ponderosa Pine growing in 3 different sites. So Arizona, California, and Oregon. And this is just a precipitation figure showing you that in Arizona, you get these monsoon rains that come through. So there's quite a bit of input in the form of rain in Arizona compared to more Mediterranean climates of California and Oregon. What they did was they used isotopes of late wood cellulose to show that these trees growing in Arizona could take up rain when it was available. And so here's the summer rain as a percentage of total precipitation. And here's the oxygen isotope. And so here is Oregon and California. That didn't really show any sort of relationship. But here's Arizona. So what they're finding is as the total precipitation increased as a result of summer rain, then the valley became more enriched. And earlier I was showing you, snow melt has a more negative signature than monsoon rain. So this is monsoon rain signal that's being captured into the cellulose of the Ponderosa Pine. So this is showing that, Ponderosa Pine have the ability to use rain if it is available. And so I went into this project thinking, well, I wonder how these trees will respond to rain events. And so I decided to take some sap flow measurements and constructed these sap flow sensors, which I'll just talk about now because Nate and Justin, my 2 students, have built many of these this past year. This is a very common way to measure transpiration in trees. This is called the heat pulse method. Basically, you have 2 thermocouples and a heater in between the 2. And the thermocouples just take temperature readings. But basically what these thermocouples do is that they take a reading, and then a pulse of heat is released. And then they take a second temperature reading. And then it's looking at the rate of change before and after this pulse of heat. So if you have water transpiring very quickly going downstream, then this thermocouple is going to pick up a greater change in temperature than this thermocouple following the pulse of heat. If there's no transpiration, then heat is going to dissipate evenly in both directions, and both thermocouples are going to sense the same change in heat. So you can use this to relate how quickly water is moving through the tree. And so here is just the equation for the heat pulse velocity that I was measuring. So I installed these in a bunch of Ponderosa Pine trees. And this is what I found. So this is heat pulse velocity. So it's not true transpiration, but gives you some idea of how quickly water is moving through the tree versus soil moisture. What you see is that transpiration is really sensitive to soil moisture changes. And so here, this light blue space, this is the upper 5 centimeters. So these are not super large rain events, but even right here, the small rain event, you can see an increase in transpiration. It's really sensitive, and not until this is about July 20th that the monsoon came, really saturate the soils, and you see transpiration rates are extremely elevated. And as you continue, the upper soils dry down, but the deeper soils are recharged, which is the reason why they're able to sustain such high transpiration rates late in the growing season. So this is telling me something about how quickly they could respond to rain, but I want to use isotopes then to see if I could pinpoint exactly where they were getting their water from. And so this is work with Max Berkelhammer, who was a postdoc with David Noon at CU Boulder at the time. And they had a Picaro out at the site measuring water vapor. So they had water vapor throughout the entire growing season. And we also collected stems. So we have June and July stems and August and September stems. And what you're seeing here is that June, July stems, this was before the monsoon rains came, the values are a lot more negative, so reflecting more of a snowmelt signature. Whereas August and September, the xylem water is right on this precipitation value. So these stems are using precip that's falling during the monsoon period. These are needle values that we also extracted, and needles tend to fall off this line because they're more enriched. Because as water is transpired through the leaves, the light isotopes are removed and the heavy isotopes are left behind. And so these leaf values tend to be a lot more enriched. You can see there's this shift going from winter rainwater to monsoon rains. And so both the isotope data and the sap flow data suggest that in this ecosystem, these trees are using rain. Okay, so this is now leading me to the third part of my talk. And before I say too much, I just want to acknowledge the summer field crew and people who were really instrumental in getting this project off the ground. So this is Kelsey, who I mentioned is a new assistant professor at University of Montana. And before I came to Montana, Kelsey and I had talked on the phone about this project. And it was because I worked in Niwot, and then I worked in Manitou. And these two sites were kind of far apart. What I really wanted to do was have a site that I could look at this sort of elevational change in one area, potentially same species of trees, different species of trees, but just somewhere where I had this continuous elevational gradient that I could test some of these theories. The other thing is, when I was working in Niwot, a lot of these modeling papers were suggesting that if snowpack decreased, you would get this significant decrease in productivity in high elevation ecosystems. But then when I was working at Manitou, I realized that these Ponderosa Pine forests are actually quite responsive to rain. And so it got me thinking, if there is a decrease in productivity as a result of snowpack decrease in high elevation ecosystems, could lower montane ecosystems sort of make up for this difference? Because a lot of these climate models are predicting more precipitation just in the form of rain. So could Ponderosa Pine use more of this rain and increase productivity? And so I was really looking for a site that allowed me to test this along an elevational gradient. And then when I talked to Kelsey, who started working at Lubrecht, it was like, aha, this is the perfect place to test this. And so Kelsey and I started collaborating even before I arrived in Montana. And when I came here, we sort of started with a sprint, essentially. And then Nate and Justin came on board in June and were just really great in building the sap flow sensors and getting the site set up for this summer. And this is Zach, who is Kelsey's master's student. So we were out in the field a lot this summer getting the site set up. Okay, so Kelsey and I talked a lot, and we sort of came up with 3 questions that we really wanted to address. So the first question we had was, how does landscape structure mediate the spatial pattern of water delivery, storage, and availability across the watershed? So like I was saying before, I was sort of expanding my ideas into taking landscape structure into account and not just thinking about how the trees use water, but what controls how water reaches the plants in the first place. How does a convergent or divergent slope determine how water stays in that landscape? So then the second question is, so once that water is there, what are the resulting true responses to these spatial and temporal patterns of water availability under different precipitation regimes? Okay, so once that water is there, how are the plants using water in these different areas? And then 3, how does the vegetation mediate the connectivity of subsurface hydrologic flow paths to the stream network? So try to think of this in a feedback. So how does the landscape affect the tree? And then how do the trees then affect what's going on in the landscape? So we have not addressed any of these questions, but we are working towards addressing all of them starting this summer. Okay, so to sort of get at question 1, how does landscape structure affect water, mediate water through the landscape? This is just some work that Kelsey did for his PhD here at Montana State. This was done at the tenderfoot experimental forest. Okay, yeah. And so what he was looking at is the connectivity between hill slopes and riparian areas. If I get this wrong, Rob, you can correct me on this figure. So he was looking at how connected, if you have a hill slope, is the watershed to the riparian area. And so he was measuring the water table of the riparian area in the hill slope. And what you're seeing here is that there's really no connectivity until snow melt, and then another pretty large rain event. And so in this figure, for the entire year, there's only about 8% of hydrological connectivity. Okay, so the water is connected from the hill slope to the riparian areas. But what he found was that this connectivity, so what percentage of the year this area is connected, is a function of UAA, which is upslope accumulation area, which Tim has talked about. But it's a measure of how much area water has potentially to accumulate. And so if you have a larger UAA, then your percent of connectivity increases. And so he took this data and applied it to the entire watershed. And the height here represents the amount of connectivity. So higher means more connected, and the different colors just mean different aspects. So this is just showing you how sort of topography can mediate how water is connected within the hill slope, but also areas where water tends to accumulate in the hill slope. So this is we're looking at landscape controls of water availability, but we also know that plants are taking water out of this landscape. So what happens when you have plants drawing this water out? How does that affect the connectivity then between the hill slope and the stream? And so this is why Kelsey and I work together, because you need someone who can come in and look at the plant water use component of this research project. Okay, so this is the Lubrecht experimental forest watershed that we're working in. This is the North Fork of the Elk Creek. I'll explain a little bit what these little dots are, but this is a Elk Creek that's going up. It's a watershed that runs west and east, and it's pretty ideal because it allows us to establish plots that are north and south facing. And so this summer we went out and we established 4 plots. This is a Google Earth image showing this. So we have established 2 plots that were lower in elevation, so a north aspect one and a south aspect, and we went up higher elevation, did the same. So we've got 4 different sites, and the idea was that we wanted to have a large range of soil moisture, vegetation types, solar insolation. And so in all 4 sites we have Douglas fir, but this other species do differ. So in the higher sites we have Engelmann spruce, which we don't have down here. And the idea that Engelmann spruce, from my previous work, suggests that they're really restricted to snowmelt and may not be as responsive to rain. Whereas down here we've got Ponderosa Pine, and not up there. We know that Ponderosa Pine down here tend to use a lot more rain. And so we've got different species that might use water differently, but then we also have one species, which is Doug fir, that lives in both sites. And so we're hoping that somewhere, in this area, we'll see a shift between snow and rain use in the different plant species. Okay, so what I really wanted to address this summer and start taking measurements on was the second question. What are the resulting true responses to these spatial and temporal patterns of water availability? Okay, so sort of a subset of that question, I want to know what are sort of the long-term responses and what are some of the short-term responses? So interannual versus within a season. So this is Zach, Kelsey's master's student. He went out this summer and he cored 520 trees in the watershed. So each of one of these red dots is where he cored a tree. So he cored a ton of different species, large Ponderosa pine, Douglas fir, Engelmann spruce. He was trying to get a range of landscape positions, and he did both north and south aspect. And he was trying to relate productivity to growth. So he was looking at interannual sort of long-term responses to where the landscape position is. And so he took these tree cores, and this is very preliminary data, but he's been looking at relative growth rates using different programs, and then trying to relate that to changes in, this is precipitation in snow water equivalent, the snowpack data that he has. And it's taken a while because he has to mount all these and scan all these samples, so it's taken him quite some time to get through them, but he has a little bit of preliminary data. And so this is data that he's nice enough to share with me. This is a topographic wetness index. So this is sort of a proxy. It's a static value, but it's a proxy for how much potential, what the potential for water to accumulate in that area is. So if you have a high TWI, probably a wet area, low TWI, probably pretty dry. So what he's finding is that as you increase your TWI, the annual growth increases. So for both Ponderosa pine and Douglas fir. So, I mean, I'm sure a lot of you would say, of course, right? If you're more wet, you probably can grow more. But this is really interesting. So Nate, I don't want to steal your research ideas, but Nate has this great idea that, this tells you something about how much they grow, but it doesn't tell you how vulnerable these trees may be to future climate. So if you're growing in a very wet environment, you're going to be growing so that you have very large vessels. So if there's drought, you might actually be more, you might be less resilient to drought. Versus a tree that's grown here, you might build your architecture so you can stand very high tensions. And so if there is a drought, you might actually be less vulnerable to drought. So that's something that Nate will be exploring. Okay, so what are some of the plant responses within a growing season then? So we went out and we put a lot of instruments out. So this is Justin in a nice pit. He's putting in soil moisture probes. We put them in at all 4 of our sites. We put them in at 10 centimeters, 30 centimeters, and 50 centimeters. And then we also installed a met station at each of these sites. So we've got a rain gauge, a relative humidity and temperature sensor, and soil moisture. So at each one of these sites, we also have it split up into like subplots. We have a plot in the hollow and a plot in the side slope. So we're looking at how landscape position at 4 different, two elevations, 2 aspects, but then side slope and hollow. So we're trying to cover all the range of soil moisture and meteorological conditions that we think we'll encounter. And here's one of our sites that we've had to barbed wire because of animals. There's a nice picture of Rob, who came with us on our most recent field trip. Okay, so here's just some data that we have from all the 4 sites. I mean, this is not data that's going to get analyzed and written into a paper, but I just wanted to show you sort of the differences that we see. We have precipitation, air temperature, and vapor pressure deficit. And so this is the southern aspect, higher elevation, southern aspect, lower elevation, north aspect, high elevation, and north aspect, low elevation. And what you see is that the differences aren't too great. So if you look at air temperature, if you look at the pattern between sort of the north and the south aspect, the range for the north is slightly less, but it's really not that different. The maximums, they all go above 30 degrees, and it's not that much colder in the north aspect compared to the south. And same with VPD, the magnitude is slightly less, but not too different. So even though VPD and air temperature aren't that different, it's really the soil moisture regime that is quite different between these sites. Okay, so again, we've got south aspect, high elevation, and low elevation. And so I just want to walk you through one of these figures to what you're seeing. So these sort of neon colors are traces of soil moisture on the hill slope, while these darker traces are in the hollow. And so at one of our driest sites, the low elevation southern aspect, you see in the hill slope, it's just really dry all year long, or all summer long, I should say. Whereas the hollow, you see some response to rain inputs, and the deeper soils don't change that much. Now you contrast this to the higher elevation southern aspect still, you see that it's a lot more dynamic, and the hollow and the side slope aren't too different from each other, okay? So the neon colors are from the side slopes. I'm sorry, the scale is kind of small. But then you look at the northern aspect, it's really wet here. So here's the slope value. So the slope on the northern aspect is almost the same as the hollow in the southern aspect. So there's a lot of variability in soil moisture regime happening in these 4 sites. Okay, I just had to show you this. So we caught this on a critter cam because we had this these sensors here, and something kept unplugging it. And so every time we would go out, and 2 sensors would be unplugged. And so Justin and Nate built this barbed wire fence. Something still got in there. So we put out a critter cam. We thought we just have to see what's coming through. And so we saw a couple of deer, and then this guy came into the frame, and we all shouted because it was so cool. But I'm just envisioning this cat like pawing at the sensors. So we have the cam going now. So next time we go out, we'll download and see what other animals we might see at our site. And this is 7:30 in the morning. We came here like 4 days after, around 8 o'clock in the morning, to download the data. So kind of gave us the creeps a little bit, knowing that this animal was there so close. Okay. So that's why there was some missing data in the previous figure. Blame it on this guy here. Okay.
No Yetis?
Not yet. I don't know. We'll see. Okay, so Justin and Nate came in June, and right away they started working on building these snap flow sensors. They worked really hard, and we were able to get these out in the field in August. But we only decided to put snap flow sensors in the southern aspects because these sensors tend to break over the winter, and we wanted to start next year fresh. So around April, I'm putting all the different sites, and so we put these out in the southern aspect, and we put a solar panel, so we can leave these in the winter and see what happens. So this is just a data logger box. Justin and Nate working hard. Okay, so here's some of the patterns that we saw with sap flow. So here's the sap flow velocity. This is contrasting a hollow versus a slope. And this is for Pinus ponderosa, so Ponderosa Pine, the hollow and the slope. And there's quite a bit of missing data. We had some issues with the site. We had one problem where some people went and opened up our boxes and just left the data logger open, and it got rained on, so we missed a lot of data. And so a lot of people come through this area, and so we're working on how to people-proof our site a little bit. But hopefully next year we can have a much clearer picture of transpiration. But you do see some differences, between the hollow and the slope. The magnitude is quite different. Right here is just the DVH of the trees to give you an idea how different the trees were in size. This is Ponderosa Pine. This is Doug fir. Again, there seems to be some difference between trees growing in the hollow and trees going in the slope. Okay, so now we go higher elevation, and now we have Engelmann spruce, and you see that the rates are actually a lot higher. So I had taken sap flow measurements in Niwot Ridge as well, and Engelmann spruce have really high transpiration rates, and that's because they tend to grow in areas where there's a lot of water. So they can sustain relatively high transpiration rates because they're not really water limited. And so you see that here in the hollow, really high fluxes, a little bit less when they're growing on the hill slope. And this is Doug fir, again, slightly higher in the hollow and less in the slope. Okay, so the other thing I want you to take away from these two figures is that there's quite a bit of variability in these flux rates. Okay, so for example, here you've got a tree that's 53 centimeters in dBH having lower flux rates than a tree that's only 19 centimeters in dBH. Whereas for Douglas fir, in the slope, you've got this dark maroon tree, which is much larger, 48 centimeters, having very similar fluxes to a tree that's only 22 centimeters. Now, this is not total transpiration, so once we actually calculate total transpiration, these values might change, but the fluxes aren't different. This just gets at this idea that there's a ton of variability in sap flow rates. And to be able to capture sap flow across the landscape is really difficult. You know, from the previous picture, you see we're really limited to where we can actually put a sensor in because of the wiring. So we have a data logger, and we can't go too far. And so in the landscape, you're trying to get the variability. You know, you want to be able to choose whichever tree you want. You don't want to only be able to choose the trees that are 15 meters away from your data logger box. So this is sort of a limitation that we are currently struggling with, is how do you measure sap flow across the entire watershed in a costly manner? I mean, if you had infinite amount of money, you could do it, but you can't yet. So the other thing that we did this summer was at each site, so at all 4 sites, and then in the hollow and the side slope, we collected isotopes. We collected branches or wood cores from the dominant tree species. We collected soil profiles down to 45 centimeters, rainfall, and stream flow when it was present. And so we've got about almost 1,000 samples that we collected this summer that we need to analyze, and hopefully it'll tell us something about water use. And I'm hoping, showing something that happens between these two different elevations where snow versus rain, the importance of snow versus rain change. Okay, so next step. We need to analyze our samples. Before we do that, I need my lab. So Dave's not looking at you for any reason except to say that it would be nice to start getting things going.
Erin says the door will be done tomorrow.
Yeah, so we have all of our samples backed up. We're going to get an extraction line set up. Like I said, over 1,000, almost 1,000 samples to extract. But then we'll analyze that on our Picaro here. And then the other thing I'm really keen on doing is to take those wood cores that Zach had collected and do some isotope work. So use oxygen and hydrogen isotopes of the cellulose to look at historical water use. So you can sort of partition rain versus snow looking at cellulose as well. So we get a historical idea of, get a historical trend of water use. And maybe there's some periods of drought that, this, like Doug fir, for example, may have relied more on rain than in snow, or even Ponderosa Pine may have used more snow than rain, or anything. We don't know. And so I'd really love to do some more isotope work, and then also looking at how landscape position affects that. And so this is something that Kelsey and I have been working on writing into an NSF grant and getting Greg to help us with this sort of analyses as well. And then finally, developing some sort of wireless sap flow. Like I was saying, we're really limited to how far we can go. And if we can do something that's wirelessly, we can just go into the landscape, choose a tree, install a sap flow sensor, and account for some of this variability. And so this is a sap flow wireless probe that's built by this company in Australia, ICT. It is ridiculously expensive. It's like $10,000 for like one little set. And so Nate is really keen on working with these cheaper microcontrollers to do the exact same thing. And so this is what we're hoping we can get accomplished this winter, and hopefully next summer we can put some out in the field and test it. Okay, so questions?
do you know how the different age classes of trees respond, preferentially or out of necessity, use rain versus snow? I mean, because that would presumably affect them at different times.
It's a really good question. So the great thing about working at Lubrecht is it's a managed forest. And the watershed that we were working in, they were all logged relatively recently, and so they're all, like 70 to 100 years old. So presumably by then, the roots have been established, and so you shouldn't see a huge difference in trees that are 70 versus 100 years old. But yes, if they were, a seed.
Do you know from like your previous work down in Colorado?
No.
Okay.
I mean, I can make some inferences on what I would.
I was just curious.
But yeah. And I don't have any isotope. We don't have any isotope data from Lubrecht, so I don't even know what the water isotope data look like yet.
You measure sap flow over such a small distance on a tree. How representative of that is that of the sap flow of the whole tree?
Yeah, well, very good question. So these sensors have two depths inside, so we're measuring traces closer to the outside and slightly more inside. So we see what the decrease is, and there's been a lot of published papers that show how sap flow decreases as you go into the tree. So the way we do our transpiration calculations is we from the DBH, we can calculate how many rings there are, and then we figure out, each ring is multiplied by the sap flux from that particular ring. And so it's going to decrease as you go into the tree, and so that area is multiplied by the decrease in sap flux as you go inside. Nate's cored a bunch of trees this summer to sort of get at how deep do we need to go. Some trees have a pretty obvious heartwood and some don't. So the trees that have a heartwood, you can stop counting the rings when you get to the heartwood. For those that don't, we have to go in a little bit further and make some inferences.
Is sap flow the same all the way up the tree?
You do it at, we put these in at diameter breast height to try to standardize it. It shouldn't change too much, but yeah, if you're at the top of the tree, it might be different. But it's also just the heat flux itself might not be different, but you'd be multiplying it by a different ring width, yeah.
It seems like it might be challenging to relate your soil moisture measurements to where the tree roots are actually accessing moisture. I wonder how you're thinking about that and how the root systems of these species might vary.
Yeah, so if you try to do some relationship between sap flow and soil moisture, you get a horrible relationship, right? Because plants, they can transpire to some potential pretty well, and then it kind of drops dramatically. So we're not trying to correlate the soil moisture necessarily to the sap flow rates. I think the isotopes will help us figure out where along the profile they might be getting some of the water from. But if we're trying to look at the landscape, it's more just an idea of if more water is available there and it's in the form of snow, we should be able to see that in the trees and the isotopes throughout the season. And I didn't show the figure, but we do, there is a nice relationship between BPD and sap flow, so that's really what drives the transpiration rate itself, not necessarily the soil moisture.
So that connectivity that Kelsey graph that you put up there, now is that connectivity water flow across the surface, or is that water flow through the profile and then like coming back out again?
Yep, it's lateral, subsurface flow.
So when you measure the isotope ratios in your soil water samples, what's the lower end of your soil moisture content that you can actually get enough water out to be
pretty low? You can get it from pretty dry soils because in that distillation it's under vacuum, so you're pulling out pulling out a lot of water in pretty dry soils. Yeah, so even the soil moisture that was down to like 3%, 5%, I'm pretty confident we'll get water out. And we don't have any soil potential sensors out there yet, but that's what we actually need, because that trees can pull water from pretty low potentials.
How much soil do you need per sample?
Not very much, like a vial, I don't know, like 2 grams. More than 10 mils, like 20 mils of soil. Probably even less, but
Had you given any consideration to studying aspen at all, where their feet are always wet?
yeah. So we don't have too many aspen in this site. I thought a lot about aspen, especially in Colorado. It's been a nice comparison looking at aspen versus the conifer, but I haven't had a chance to work with aspen. But yeah, I mean, those guys would probably just be pumping water through, and I'm sure their rates are just really high. But presumably, they would be using, snow melt and rain. I think just as equally. That's just my hypothesis. I don't actually have any data to prove that, but because they're able to maintain such high transpiration rates, they must be using all the water that they can access. And they've got that network from those clones, so they can probably access water in areas that conifers might not be able to.
I mean, I've worked on Ponderosa Pine a lot in Oregon, and one of the things we found there, we were looking at trees across the landscape of different ages as well. And when you start looking at total transpiration or evapotranspiration from the landscape, it tends to be very similar. You can go into some areas where your leaf area index is made up of very different species. So you might go into a young site where the leaf area index of the shrubs could be a significant component. When you start looking at things like that, you can find, like, for instance, the shrubs that may have been there a long time can actually use a lot of water later in the season because they've got deeper roots than the trees. So there's lots of interesting patterns like that. When you start measuring trees themselves, you have to think about what maybe other things are doing.
Yeah, no, that's a really good point. Yeah, one of the things that my students have talked about is, like, your neighbor influence. So you're a certain size, growing in a certain area, but what if you have a huge neighbor next to you who's competing with you for that same water resource, So that's something that we've talked about, how you go out and measure sort of your nearest neighbor influence, potentially.
The same on burn sites. We did a lot of work on burn sites looking at, and it's amazing how much annuals and perennials that come up a few years after a burn can contribute to the water balance.
Yeah, so we haven't.
All these different things make it very complicated when you start moving around.
I agree, which is why if we could make a wireless sensor and deploy a ton of these in the field in an economic way, we can maybe tease out some of these other potential factors that we're not considering right now. And we're really limited by how much we can do right now. We've got 12 meters. That's it. Hopefully we have a tree that's 12 meters away from the data logger box.
Just following Stephanie's question, do you have any idea or plans to dig a deep pit? Like, some of those trees look pretty big and sort of stretches my imagination. I think that root plate just sort of says, okay, 40 centimeters, I'm done.
Yeah.
Not looking.
Well, so the southern aspect of the North Fork Elk Creek drainage, Kelsey went out with an excavator pit and he dug. I don't know how much did he dig?
It's like 3 meters, I think.
Yeah, and there was still soil, so it's granitic, so there's really deep soils in that area. And Ponderosa Pine, they can grow anywhere. And their roots will just grow wherever they can, so they can grow in a rock or in a very well-drained, soil. So if you ask me how deep the roots go, I think it just depends on where they're growing. Yeah, so he dug 3 meters, and we saw little roots. Well, I didn't see them, but they saw little roots down there, so I know they go down that deep.
Do you think remote sensing will help in the future get at these questions that, I don't know a lot about it, but it seems like you're working from a tree scale and you're trying to get at transpiration, right? Do people remotely sense that, and will the technology catch up to the point where you can look at different elevations remotely in terms of just transpiration rates throughout the year?
I know people do remote sensing for ET, so total evapotranspiration. That's always the hard part, is partitioning out the two components. And so you can probably remotely sense evaporation or evapotranspiration. I know there's some methods to try to partition out, but you still need a sap flow to, I think, compare that to. And so sap flow would kind of allow you to do the scaling from ground up, bottom up scaling, and then remote sensing could do top down, and hopefully the two could, hopefully they match. What we really need is a tower at the site, but we don't have one. Yeah.
Ask Dave.
Yeah. Yeah, I'd like to amend my startup.
It's okay.
facilities do they have?
Great facilities. There's a nice lodge, a conference center, a dining hall.
Anybody can work there? How do you
Anyone
do
can
that?
work there. We've got a special deal with the forestry school, so they let us stay there for free when we go. We just have one little cabin, but you can rent out, cabins. There's great cross-country skiing. People go there a lot to do that. So
Connected to a dude ranch.
yeah, a really expensive dude ranch.
So a good field base.
Yeah, it's just a little too far. It's 3 hours, so we can't just go there for the day. But we don't really mind going there and spending the night. It's really a nice place to work. Yeah.
Well, thanks, John.